Hello, my name is Tim Bergquist. I'm a data scientist at the Icahn School of Medicine at Mount Sinai for the Institute for Genomic Health, and today I'll be talking about annotation database updates and their effects on missense variant pathogenicity predictions. Bioinformatics resources like Ensembl, UniProt, and RefSeq, as well as variant annotation tools like VEP, Anavar, DBNSFP, and OpenCravat, are continually updated with the most current genetic and molecular information. These updates may involve changes to the mappings between nucleotide variant coordinates and amino acid variant positions, the actual protein sequence itself, or the designation of a canonical status to an isoform, among many other types of information. Variant annotations are often used to map chromosomal variants to their consequent amino acid substitutions, using a given version of the backing bioinformatics resource. Here we see Ensembl's VEP annotation tool, version 96, being used to map chromosomal coordinates to RNA transcripts, genes, and the resulting amino acid substitution from the associated protein sequence. As the bioinformatics resources update, these mappings can also update and change their predicted genomic and proteomic consequences. Missense variant pathogenicity predictors, like MutPred2, typically use such protein-level information from the aforementioned bioinformatics resources to calculate pathogenicity scores in order to categorize a missense variant as either pathogenic or benign. These tools are often used in clinical classification pipelines in order to determine whether a variant is potentially involved in a disease, i.e. pathogenic, or whether it is unrelated to the disease, i.e. benign. For scalability and convenience, it is common practice for missense variant pathogenicity predictions to be pre-computed exome-wide and made available to end users, often through the previously mentioned variant annotation tools. However, such static pathogenicity predictions are not expected to be in synchrony with updates to bioinformatics databases and may give discordant predictions if the predictors were run using the latest information. Here we ask, does that matter? Do bioinformatics database updates affect missense pathogenicity predictors and their use in clinical classification? In order to get at this question, we wanted to ask, are important variants affected? So we looked at all variants that appear in clinically actionable genes, as well as in ClinVar, and we asked how many are affected by bioinformatics database updates. We looked at all genes that appear in ClinGen Actionability Knowledge Repository, which contain 296 genes. We found all ClinVar variants that appeared in those genes, resulting in 70,566 variants that met this criteria. We looked at the number of times those 70,566 variants were impacted or changed between Ensembl version 90 and Ensembl version 110. We used the VEP annotation tool from Ensembl and ran that on these 70,566 variants. And we used all versions of that tool from version 90 to 110 to get all the different potential annotations for these variants. We asked the question, do variants remain unchanged across these versions? The blue part of the flowchart is the number of variants that remain unchanged across all versions. As variants change with updates to the database, they get paired off into these orange workflows, into these orange flowcharts. Of the original 70,000, 59,188 variants remain unchanged in their amino acid substitution. 9,000 have at least one amino acid substitution change across all versions. 2,236 variants were changed twice, and 79 had 3 changes across all versions from version 90 to 110. But then the question is, okay, so what? Are these clinically impactful? In order to get at this question, we're again here looking at this chart. We're again looking at changes across versions of Ensembl's VEP. But here we have scored with MutPred2 all 70,566 variants with a score between 0 and 1. We have scored all the variants across all versions of Ensembl between 90 and 110, and we're looking at the impact that Ensembl database updates have on the MutPred2 score. We have mapped the MutPred2 scores to ISMB AMP clinical evidentiary strengths in order to classify them as whether they as either supporting pathogenic PP3, indeterminate, or supporting benign BP4, using a calibration technique from the manuscript reference at the bottom of the slide. As we walk across the ensemble versions, we see from 91 to 92, 459 variants changed from their original clinical classification to a new classification, either PP3, indeterminate, or BP4. From version 95 to 96, 2,262 variants had their strengths of evidence changed to be either supporting PP3 or supporting benign or indeterminate. And then from version 101 to 102, 2,486 variants changed in their clinical classification evidence strength. Cumulatively, across all versions from 90 to 110, 63,498 remain unchanged, but 1,839 changed to a strength of evidence for PP3, or at least supporting pathogenic, when they previously did not have that strength of evidence. 3,437 variants changed indeterminate, and 1,792 changed in their strength of evidence to benign. Changes in annotation databases and their interfacing software can have direct effects on the clinical interpretation of clinically relevant missense variants. While the percent of total affected variants in this study was approximately 10%, these changes only represent a small subset of missense variants. Again, we only looked at 296 genes, and we have only looked at a singular bioinformatics resource database. Different bioinformatics databases or prediction tools may be differently impacted. As future ISMB AMP guidelines envision a more prominent role for variant impact predictors in clinical variant classification, our results have direct implications for their proper incorporation into clinical workflows. Our preliminary recommendations include explicit use of sequence version numbers and annual updates to bioinformatics pipelines for clinical variant interpretation. Thank you very much.